Welcome to part 24 of this video on the immune system. Okay, so we're currently studying the humoral adaptive immune response, and the point that we've got up to at the moment is the production of a B cell germinal center. And we've seen what's going to be happening inside this B cell germinal center. We've seen the process of somatic hypermutation, class switching, and we've discussed that affinity maturation will be occurring. The population of cells in that germinal center will gradually be evolving so that the cells overall have immunoglobulin molecules with antigen binding regions that have higher affinity for the epitope of the antigen, which the humoral adaptive immune response is against. What I now want to discuss is where do the actual effector B cells come from, because the cells of a germinal center, they are not the effector B cells. The cells of the germinal center are known as the germinal center B cells. So let me just draw one of these germinal center B cells here. So here is its nucleus, and this is a germinal center B cell. So this will be participating in all of the activities that are going on inside the germinal center. However, it will not be producing secreted immunoglobulin molecules, which is the characteristic of effector B cells. Those, of course, are the antibody molecules that are actually going to go and help clear the pathogen. So this is a germinal center B cell here, and I'll just color it in red. So we want to now discuss, where do the actual effector B cells come from? How does the germinal center result in the production of effector B cells? So, at the moment what we've seen is that the germinal center will be producing germinal center B cells that have immunoglobulin molecule designs that have a greater and greater affinity for the epitope of the antigen. Now, what happens is the germinal center B cells, they can eventually decide that they're going to start differentiating into either an effector B cell or a memory B cell, and it's not really understood when one of these germinal center B cells actually decides that it's gone through enough rounds of affinity maturation and it's ready to actually differentiate into either an effector B cell or a memory B cell. So the trigger for differentiation isn't understood, but the germinal center B cells will eventually differentiate into effector B cells or memory B cells. So I'll state it again because it's really important. We don't know what decides when a germinal center B cell is ready to actually differentiate, but something does determine that. And germinal center B cells, they will eventually start to differentiate into either an effector B cell or a memory B cell. So let's say we've got a germinal center B cell here. What it can now do is it can differentiate into either an effector B cell or a memory B cell. So let me just draw pictures of these. So firstly, let's just discuss the effector B cells. Effector B cells have another name which is in common use. They're also known as plasma cells. So an effector B cell can also be called a plasma cell. Now, these have a very different shape to the original B cells. They have a sort of oval shape rather than a round shape. B cells that aren't effector B cells, they have this very characteristic round shape where the nucleus dominates the cytoplasm. In fact, this picture is probably a little bit incorrect. The nucleus should be more this sort of size here. The nucleus dominates the cytoplasm of the cell, whereas effector B cells, they have an oval shape and they have a much larger cytoplasm to nucleus ratio, and this cytoplasm is going to be where all of the secreted antibody molecules are actually going to be made, because that's the characteristic thing that effector B cells do. They are actually going to produce the secreted form of the immunoglobulin molecule and put that into the bloodstream, and therefore it will go off and have the anti-pathogen effect. So these are the cells that are actually going to contribute to the eradication of the pathogen. So here in green, this is an effector B cell. Now, if the germinal center B cell that produces this effector B cell, or the plasma cell, had undergone class switching, i.e. it had changed the immunoglobulin type that it was making, then the effector B cell will also produce that same class of immunoglobulin. So if we bring back up this picture, if the germinal center B cell, for instance, had class switched, and now the immunoglobulin heavy chains that it produces are of the IgG2 type, then of course when it produces secreted immunoglobulins, all it's going to do is now it will splice out the portion at the end of the constant gene segment which pertains to the membrane implanting portion of the immunoglobulin heavy chain, but it won't change suddenly which gene segment it's using for the constant region of the immunoglobulin heavy chain. That will remain exactly the same, so it will still produce the same class of immunoglobulin. So whatever class of immunoglobulin molecule that the germinal center B cell was producing, the effector B cell that it differentiates into will also produce that same class of immunoglobulin molecule, except that now it will be the secreted form rather than the membrane-bound form. Okay, so we'll come back to talking about effector B cells in just a moment. Let me just mention memory B cells. So this is a similar principle to memory T cells. What we want to do is we want to create cells that are effectively equivalent to the naive, mature B cells, so that we are ready in case a second round of this pathogen ever comes back. And these are the memory B cells. So memory B cells, they will do exactly what mature naive B cells do. They'll just circulate between the circulatory system and the lymphatic system, and they can be activated in the same way that the mature naive B cells can be activated. The difference is that you will have a much bigger population of these memory B cells than you had originally in the primary adaptive immune response of the mature naive B cells. So in the primary adaptive immune response, that was the first time you'd ever been infected by this pathogen, then you had very few of those mature naive B cells where the immunoglobulin molecule was targeted against one of the epitopes of one of the antigens of this pathogen. Now, on the secondary adaptive immune response, when you're infected the second time, you'll have a big population of memory B cells, and that means that we can activate the humoral adaptive immune response much quicker because it's going to take much less time for one of the antigen molecules or a fragment of the pathogen with lots of the antigen molecules on to actually reach one of these memory B cells and therefore activate those memory B cells. So let me just draw a picture of a memory B cell. So these look more like the classical picture of a B cell where we have a very large nucleus, very little cytoplasm, like so. Because of course these aren't going to be particularly active, and they, like the mature naive B cells, will only produce membrane-bound immunoglobulin. The difference between these and naive B cells is that the naive B cells, remember, they could only produce immunoglobulin molecules of the IgD or IgM type. This memory B cell will produce the same type of immunoglobulin molecules as whatever the germinal center B cell, which produced it, produced. So if this germinal center B cell had undergone class switching and now produced IgG2 B cell receptors, then the memory B cell will also now produce IgG2 B cell receptors. So they stick with the same class of immunoglobulin molecule as the germinal center B cell which produce them. So that's something different between the naive, mature B cells and the memory B cells. The other thing to say about the memory B cells is that they have a much larger lifespan than the mature naive B cells. Again, they can live for the entire life of the organism. So these can stay with you for the lifetime. Okay, so lifespan, your lifetime. Right. So that's memory B cells, and they, like the memory T cells, are responsible for the difference between the primary adaptive immune response and the secondary adaptive immune response. Okay, I'm not going to say anything more about the memory B cells. Now what we're going to turn our attention to is the effector B cells, the plasma cells. So, overall, our germinal center will start chucking out effector B cells, plasma cells, which are these germinal center B cells that have differentiated and are now going to start producing secreted immunoglobulin molecules. And the major form of secreted immunoglobulin molecules that most plasma cells produce is the IgG form. Okay, so let me just firstly tell you the story of what's going to happen to the effector B cells, and then I'll come back to discuss what the secreted immunoglobulin molecules, the antibodies, are going to do. or at least remind you of lots of the things that we have already said that the antibody molecules are going to do, because we actually have talked about most of the things that antibody molecules can do already in the video, but I'll summarise it again. Okay, so what's the story then for the effector B cells? So we're producing them at the site of the germinal centre. Germinal centre B cells are gradually becoming effector B cells, and these of course will have an immunoglobulin design that is very good and has a very high affinity for binding to that epitope of the antigen. They're going to go into the bloodstream gradually, and we've discussed how you can go from the lymphatic system back into the bloodstream. So they're going to get into the bloodstream, and then they're actually going to go and live in the bone marrow majorly. I mean, some of the effector B cells will just circulate in the bloodstream, but the major place that effector B cells go is to the bone marrow. So let me just draw a picture of this here. So effector B cells will go back to the bone marrow. So we have the story returning to the bone marrow. So here again is my picture of the femur, like so, the shaft of the femur here, and then of course the bone marrow is inside the hollow portion of the bone in the middle of the bone shaft here. So effector B cells go back to the bone marrow, and they can live in the bone marrow for an incredibly long time. They can stay there for up to a year about after the pathogen has been cleared, so after the immune response has terminated, and that's why you will continue to find secreted immunoglobulin molecules, antibodies directed against the pathogen in the blood for, you know, up to a year after you've actually had the infection. Okay, so effector B cells will go to the bone marrow, and there they will start chucking out secreted immunoglobulin molecules. And as I say, the major type of secreted immunoglobulin molecule that most of the effector B cells produce is IgG type. So IgG1, IgG2, IgG3 or IgG4 immunoglobulin molecules. So the effector B cells go to the bone marrow and they start chucking out immunoglobulin molecules. The immunoglobulin molecules will go into the bloodstream, and then they will leave the bloodstream at the site of infection. So they are proteins that can exit the bloodstream at sites of infection with the inflammatory exudate. So this is another major reason that you want to form inflammatory exudate at the site of an infection, because it means that immunoglobulin molecules that your effector B cells are producing can leave the bloodstream and go into the tissue fluid. So you're going to get immunoglobulin molecules coming to the site of infection. So I'll just show this here. So the bone marrow, from the bone marrow you're going to get loads of immunoglobulin molecules, and then these are going to go to the site of infection. They're going to leave the microvasculature that is they're going to leave by the microvasculature that is undergoing the inflammatory response. So throughout this video we've often used the example of the infection being present within the skin. So here they'd come into the tissue fluid at the skin, and then they're going to help with the fight against the pathogen. And what I now want to discuss is how are they going to help with the fight against the pathogen, and in particular I want to ask, how do the different types of immunoglobulin molecule differ in how they help fight the pathogen. So let's start off with things that absolutely all of the immunoglobulin molecules are fantastic for doing. And the thing that they're absolutely all fantastic at doing is they bind to the antigen molecule that they are directed against. So if I just show one of these secreted immunoglobulin molecules, here is the antibody, as we can now call it here. It will bind to the antigen that it's targeted against at that specific epitope that it's capable of recognizing. And the thing that they are all capable of doing is destroying the function of whatever this protein is. So this antigen, it might have some very important function for the pathogen, and the binding of an immunoglobulin molecule might disturb that function. So for instance, here's some examples. What if this protein that we've launched this adaptive immune response against was a receptor on the surface of the pathogen that the pathogen relied upon for, let's say, getting inside a cell? So let's say this was an intracellular parasite pathogen. Let's say it likes to go inside of human cells. If this antigen that we've launched this humoral adaptive immune response against is the receptor by which the pathogen actually gets into cells, then you can imagine that the binding of the antibody molecule to that protein is going to stop it being able to bind to whatever its target on human cells is, and therefore we stop the intracellular pathogen being able to enter cells. So there's an example of why it might be really good just to have the immunoglobulin molecule bind to the antigen and disable its function. In addition, this protein might have been some sort of secreted toxin, so the antigen that we've launched the humoral adaptive immune response against might be some secreted toxin that the pathogen releases and which kills human cells. Again, if you bind an immunoglobulin molecule to that toxin, then you might disable its function. So that's something that absolutely all of the different classes of immunoglobulin molecules are capable of doing, binding to the antigen, whatever it is, and disabling its function. And that might be one of the really valuable things that they do, and they're all capable of doing that. Okay, what I now want to talk about is the other functions that immunoglobulin molecules are capable of doing, and these other functions are going to differ for the different types of immunoglobulin molecules. So I'm now going to go through what the 5 different classes of immunoglobulin molecules are actually good for doing. Okay, so we'll start off with IgD. So the first thing to say is IgD is not produced in the secreted form. So none of your effector B cells will be producing IgD immunoglobulin molecules. IgD is only used as a membrane-bound immunoglobulin, so it's only ever used as a B cell receptor. So none of the effector B cells will be producing secreted IgD. If they have not undergone class switching, i.e. if the germinal center B cell which produced the effector B cell had not undergone class switching, then of course this will still be what the gene for the immunoglobulin heavy chain looks like, and therefore this will be what's transcribed, and when they're actually producing secreted immunoglobulin molecules, they will be using the constant mu gene segment rather than the constant delta gene segment, and they'll be splicing out the portion at the end there that codes for the membrane-bound portion. So IgD is not actually produced as a secreted immunoglobulin. So actually it's the only one where I can't even say that it's useful for neutralizing proteins and making sure that they can't actually perform their function, because it isn't used as an antibody in that way. It's just used as a B cell receptor. So IgD, it's the only one that you can only produce in the membrane-bound form, and you don't ever produce it in the secreted form. You don't ever splice out the portion of the DNA for the constant delta gene segment that actually codes for that membrane implanting portion of the immunoglobulin heavy chain. Okay, so that's IgD. Let's now go on to one that can actually be produced in the secreted form, and all the others can be produced in the secreted form. Let's now go on to IgM. Okay, so IgM, secreted IgM. So secreted IgM immunoglobulin molecules, these tend to actually form pentamers. So IgM molecules tend to form pentamers. So let me just show this with a picture. So to show this simply, I'll show each of the IgM immunoglobulin molecules as a Y, like so, and they tend to bind together to form a pentamer, like so. So if you've got an effector B cell that produces IgM type immunoglobulin molecules of the secreted form, it tends to bind 5 of these together to make a pentamer, and then it secretes a pentamer like so. Okay? So this is the way that IgM tends to be secreted. Now, what is IgM extremely good for doing? Well, it can bind to a lot of separate pathogens. This is what IgM is fantastic for doing. Because it forms this pentamer in this way, you can imagine that it's now got 10 different binding sites for the antigen molecule. So let's imagine that the antigen molecule is a surface antigen molecule. You can imagine that potentially it could be binding to 10 different pathogens all at once. And therefore it can aggregate pathogens together. And why is this so fantastic? Well, one, IgM molecules with their constant regions of the heavy chain are able to activate the classical pathway of the complement cascade. So the constant region of the heavy chain of an IgM secreted antibody molecule can activate the classical complement cascade, i.e. it can bind C1 proteins, and then the C1 protein will activate the classical complement cascade, and therefore we'll get the classical complement cascade activated near a whole bunch of pathogens, and hopefully it will help kill all of them. So the classical complement cascade is one thing, and secondly, IgM is extremely good for opsonization. And remember, opsonization means promoting phagocytosis. So again, macrophages and neutrophils, they will have receptors on their surface which can bind to the constant region of the IgM heavy chain, and this is especially fantastic because of the fact that they form pentamers, and they could have a whole bunch of pathogens clustered together, and therefore the phagocyte might be able to phagocytose all of those pathogens that are clustered together all in one go. So if I just draw this, you might have one pathogen bound here, one pathogen bound here, one pathogen bound here, one pathogen bound here, another one may be bound here, and of course they'd all be bound by their antigen molecules, but it clusters pathogens together and then can activate these two different ways of killing these pathogens, either by phagocytosis or by activating the classical complement cascade near them all, and thereby activating their destruction. So it's very good for killing pathogens, IgM. Okay, so that's the special feature of IgM. Let's now go on to IgG. So remember, there are 4 different forms of IgG, but we're discussing all of them together because they all have reasonably similar functions. So this covers IgG1, IgG2, IgG3 and IgG4. These are secreted as monomers, so just a little Y there, and they can do the things that IgM antibodies can do as well. They can activate the classical complement cascade, and they're also incredibly good at opsonizing pathogens. So they're very good at binding to surface antigens on the surface of pathogens, and then activating the destruction of that pathogen through the classical arm of the complement system, and also through opsonization by promoting their phagocytosis by macrophages and neutrophils. So IgG does all of the same things as IgM, except that it doesn't form a pentamer. It just works as a monomer. And as I say, the major form of secreted immunoglobulin molecules that you produce in most humoral adaptive immune responses is IgG. Okay? So most effector B cells that are produced in most humoral adaptive immune responses will be IgG producers. Next up, let me talk about the IgE secreted immunoglobulin molecules. So again, I'm covering both of the IgE type immunoglobulin molecules, both IgE1 and IgE2. So IgE immunoglobulin molecules, these can actually be responsible for producing whole new pattern recognition receptors on the surface of sentinel cells. So on the surface, for instance, of mast cells, so let me just draw a mast cell. So remember, a mast cell was one of these sentinel cells and was really important in actually recognizing the pathogen initially in the tissue and releasing pro-inflammatory molecules to start the inflammatory response. So I remember these sentinel cells, the way in which they recognize the presence of pathogen in the tissue initially is that they have all sorts of pattern recognition receptors, which are receptors on their surface that can recognize molecules that are giveaway signs that there is a pathogen present, molecules that no human cell would ever produce in its right mind. Okay? But there is a finite number of these pattern recognition receptors that sentinel cells have available to them. So the idea behind IgE molecules is that you can actually use the immunoglobulin molecules that you've created to make new pattern recognition receptors. So on the surface of mast cells, you have a receptor which is known as the Fc epsilon R1. So let me just show this, Fc epsilon R1. So this Fc epsilon portion pertains to the fact that we are talking about IgE molecules, so the epsilon is for E, and then it's just receptor type 1. So this is the Fc epsilon R1 receptor on the surface of macrophages, now coloured in green. And this is capable of binding to the constant region of the immunoglobulin heavy chains within an IgE molecule of either IgE1 or IgE2 type. So let me just show this here. So let's say this is our IgE molecule here that we've produced as part of the humoral adaptive immune response. The constant region of the the heavy chains of the IgE immunoglobulin molecule can actually bind to the Fc epsilon R1 receptor. And now what's the point of this? You see, the antigen binding regions of this IgE molecule, they are capable of binding to the specific epitope of that antigen that's associated with the pathogen. So I'll just draw my antigen that we launched the humoral adaptive immune response against. So here is the antigen, here. Now the idea is that this structure that we've created here, this IgE molecule loaded on this Fc epsilon R1, this can now function as a pattern recognition receptor because this is now capable of activating the mast cell to produce all of the pro-inflammatory mediators and kickstart the inflammatory response, just like any of the other pattern recognition receptors. So we have effectively manufactured a new pattern recognition receptor now that is specifically targeted against this antigen of this pathogen. So it allows us to produce this new type of pattern recognition receptor on our sentinel cells. So we can expand our repertoire of molecules basically that can now activate sentinel cells to release the pro-inflammatory mediators and kickstart the acute inflammatory response. So IgE is absolutely fantastic for creating new pattern recognition receptors on the surface of mast cells and therefore expanding the number of molecules that can actually kickstart the acute inflammatory response. And those of you who know anything about asthma will know how important IgE is in asthma. There you get IgE molecules produced against things that you shouldn't get it produced against, and then that's what can then kickstart a asthma attack. When you inhale those molecules that you've had IgE produced against, the allergic molecule, it can then kickstart the acute inflammatory response because of these new pattern recognition receptors that you've produced on your mast cells, and that's what then causes the asthma attack. But I won't say anything more about that. If you're interested in that, go to the playlist on the respiratory system and I have a video on asthma in there. Okay, so the final type then of immunoglobulin molecules is IgA molecules. Now IgA molecules, these are produced as dimers. So let me just show these. So they are produced as dimers, so I'll draw two Ys like so. So here is the midway point. So we've got one IgA molecule and then another IgA molecule. They bind together and form a dimer. Okay, so IgA is also very good at opsonizing pathogens. So just like the pentamers of IgM, you can imagine that this could be binding to lots of pathogens, maybe 2, 3 or 4 pathogens together and clustering them together, and then the constant region of the IgA heavy chain is then capable of activating receptors on the surface of a phagocyte and then promoting the phagocyte wanting to phagocytose this cluster of pathogens potentially here. So it's very good for opsonization. The other key thing for you to know about IgA molecules is that it's the main form of immunoglobulin molecule that is secreted onto epithelia. So it's secreted, for instance, onto the respiratory tract epithelium. So you secrete IgA molecules in the bloodstream onto the respiratory tract epithelium. And then if you've got pathogens present on the respiratory tract epithelium, IgA can help to eliminate them. And you'll know that there are, for instance, macrophages in the alveoli of the lungs, known as alveolar macrophages, which will then be the things to actually phagocytose the pathogens that have been opsonized by the IgA molecules. So this is the major form that is secreted onto epithelia, and an example of an epithelium that IgA is secreted onto is the respiratory tract epithelium. So secreted onto epithelia. Okay, so those are all the different forms of immunoglobulin molecules. And by the way, again here, when we were discussing IgA, we were discussing both IgA1 and IgA2. So that's the difference in the functions of the different forms of secreted immunoglobulin molecules. Right, so we have almost then finished our discussion of the adaptive immune response. What I do just want to say now is a few little words about how the adaptive immune response terminates. So we've now seen the humoral adaptive immune response, which has resulted in the production of all of these immunoglobulin molecules in different ratios. And I should say again that it's still an active area of research trying to determine what determines the different ratios of production of these different types of secreted immunoglobulin, i.e. what determines what the germinal center B cells are going to class switch their immunoglobulin molecules to. So I described how in germinal centers, the germinal center B cells can undertake class switching. We are very, very interested in the things which determine which type of immunoglobulin molecule that they want to switch their class to, because that is something that matters, and in different humoral adaptive immune responses against different pathogens, the class switching occurs differently, and you produce overall more, you produce different ratios of the different types of immunoglobulin molecules. So there's still a lot of research going into what determines the ratio of production of the different types of immunoglobulin molecules. But they're all very, very good at clearing pathogens by the mechanisms that we've discussed here, by neutralizing proteins and making them dysfunctional, by activating the destruction of the pathogen through complement cascades, and through activating opsonization of the pathogen. So what I finally want to say then is a few words about the termination of the adaptive immune response. So effector T cells will die and be removed from the bloodstream within a week of the pathogen being cleared. So once the pathogen's been cleared by the adaptive immune response, effector T cells are removed from the bloodstream extremely rapidly. So cytotoxic effector T cells and helper effector T cells, they're going to be removed from the bloodstream extremely rapidly after the pathogen has actually been cleared. Effector B cells, they're not in the bloodstream, they are in the bone marrow, and they can actually remain in the bone marrow, still producing secreted immunoglobulin molecules for up to a year after the pathogen has actually been cleared. So that is why you can still see the presence of immunoglobulin molecules in the blood, which were produced against a pathogen that you might have been infected with long ago. Okay, so well done for getting to the end of this video. I hope you've learned a lot about the immune system.